One of the problems facing marine fisheries managers is the time it takes to assess the size of the populations of the fish that they're managing. It usually takes several years. And because of this, we're developing a new method, one that can be as short as just a few months, to estimate the size of fish populations out in the Gulf of Mexico. The new method is actually one that's been used for a while, but it's had a problem that kept it from being used here. And that was that it's based on identifying fish eggs that the female fish release into the water. It's easy enough to count how many eggs there are out in the water, but it's difficult to know whose eggs they are. So you need a method for identifying those eggs. They all look alike. And what we're doing is applying a method called DNA barcoding to the identification of these eggs.
Dr. Peebles and his research students go out on a ship into the Gulf of Mexico and they collect all of the free-floating fish eggs at a given site using a plankton net. We then take these fish eggs and we extract the DNA from them. Every organism contains DNA that's unique to that organism. By extracting the DNA from the individual fish eggs, we then amplify a barcoding gene. So you can think of this just like the barcode on an item at your supermarket. When we sequence this gene, we can then compare it against a database, and that tells us what species each fish egg belongs to.
So once Dr. Breitbart has identified the eggs, we can then compare those with another aspect of the fish that we measure, and that is their rate of egg production. So if we know the rate of egg production and the number of eggs that are floating out in the water, we can calculate the number of females that release those eggs. And we can do that for every species that we capture in the survey. That's a relatively rapid and could be very cost-effective method. One of the most exciting aspects of what we're doing is that there's some just basic discovery involved. When we survey fish eggs over a large area, we know that we will find new spawning grounds that people just don't know about. The fish are underwater, they spawn at night. It's very difficult to detect where they spawn, but this method is one of the best, if not the best, methods of detecting spawning in marine fish. So we're very excited about that.